I'm going to talk about ways my group has looked at glioblastoma cancer stem cells from a very out-of-the-box approach with the role of a possible viral etiology or agent. And then I'm going to talk about two investigator-initiated trials that we're doing here that have nothing to do with the virus but are more associated with different approaches to try to attack these cells without any preconceived notion of their biology, but just by blunt force, if you will. So I mentioned these three components of the talk, and I'm going to maybe spend about 10 minutes on each. So for about 15 years, I've been interested in the possible role of viral agent cytomegalovirus and glioblastomas. In 2002, after developing a bunch of very sensitive immunostaining techniques and working on in situ hybridization techniques and PCR, we found high incidence, very high incidence evidence of CMV infection in glioblastomas. And here you can see some immunostaining with monoclonal antibodies with tumor cells and adjacent normal brain on the same slide. And we looked at several different monoclonal antibodies. And then we did RNA positive and negative control in situ and DNA positive and negative control. So it was a pretty compelling case. Subsequently, a lot of papers were published that couldn't confirm this. And so I put together a small meeting and got some of the top CMV virologists in the world to come. And so they sort of went back to their institutions and approached it with a kind of a very objective eye. And one of the investigators was Robert Kaleda at the McArdle Cancer Institute. And he found that quantitative PCR and just regular PCR of glioblastomas confirmed that there was a significant association of CMV with these tumors. And this is a panel of 12 GBMs, and they could amplify various regions of the viral genome from most of the tumors. And then another collaborator who was at the same meeting, Tim Kowalik at UMass, went back and performed similar complementary studies looking at PCR of a bunch of glioblastomas, and then Western blot for some of the key proteins that we were detecting, IE1 and PP65, and glycoprotein B, which you see in about half of these tumors here. And so during this period of time, I was in California, and my laboratory was kind of focused on, assuming this is for real, what are these viral proteins doing in the tumors? And I'm just going to skip over a lot of things that we were doing, but the first kind of focus we had, we were focusing on four of the sort of hallmarks of glioblastoma biology: the loss of tumor suppressor function, activation of signaling, immune dysregulation, and then the sort of nascent field at the time of glioblastoma cancer stem cell biology. So the first interesting findings we had was when we actually added the virus to tumors, we saw dramatic activation of downstream signaling of PI3 kinase and RAS and all these signaling pathways. And so I asked Liliana in my lab to investigate receptor tyrosine kinases that might be getting activated. And on a panel of 42, we only saw one that was dramatically induced, which was PDGFR alpha, which ends up being a pretty important tyrosine kinase receptor in glioblastoma. And we could show that when we added virus to glioblastoma cells, as shown in this image here of these green cells, they would dramatically get induced to phosphorylate this receptor and activate downstream signaling pathways. And it turned out that this viral protein, glycoprotein B, that we saw in the tumors was acting as almost exactly like the growth factor PDGF itself. It would activate this receptor and phosphorylate it and activate all these downstream pathways. And we found GB on the tumor cells, suggesting that this viral protein was activating a lot of these pathways. And it turns out that now we know that this is one of the major cellular receptors for the virus. Around that same time, we also became very interested in the fact that we were having difficulty finding the virus in the tumors, but when we sorted the cells with CD133 marker, we could enhance our ability to detect the virus, and it seemed like the virus was preferentially infecting these glioblastoma cancer stem cells, which you've just heard about. And indeed, when we grew the cells in culture in neurosphere conditions without growth factors, except for a couple of growth factors to enhance these sort of neurosphere glioblastoma stem cell properties, we could immunostain the cells, here shown in green for the viral protein IE1, and it seemed to co-localize pretty consistently with markers of glioblastoma stem cells like nestin. And it also co-localized with other markers such as SOX2, EpCAM, and some other stem-like markers. So we asked the question, and we knew that the immediate early 1 viral protein was a dramatic and strong transcriptional activator that if you put it by itself into a cell, it can mutate the cell, and in addition to one other hit, can transform cells and cause them to activate cancer pathways. So we knew that this viral gene was potentially important in cancer biology. So we made an siRNA knockout gene. We could knock it out with siRNA, and interestingly, or kind of dramatically, if we grew these glioblastoma tumor stem cells derived directly from patients in the setting of control siRNA, over 72 hours they would look like this, and with control siRNA they would have a similar appearance. But if we knock down this one viral gene of IE1 in these tumor glioblastoma stem cells, there would be a dramatic reduction in tumor growth. And that was concurrent with a loss of some of these stem cell proteins like SOX2 and nestin in these clusters. So we went on to show in several other cases of different patients that we could knock down neurosphere growth, and seemingly the stem cell properties of these glioblastoma cultures by knocking out this one viral protein. And here you can see in a control siRNA experiment, the Western blot shows that the viral protein is present, but when we knock it down, it goes away. And here is a panel of transcription factors associated with cancer stem cell biology, and we had a dramatic reduction of SOX2 and some other ones. Here you can see that there was a 40 to 80% knockdown of OCT3 and 4, NANOG, SOX2, and some other transcription factors associated with with cancer stem cell survival. And we did some further studies that indicated that when we knocked down this one viral gene, we could inhibit cell cycle progression and promote differentiation of these cells, whereas they had high levels of nestin, they would decrease the nestin production and become flattened out or have increased GFAP expression. So that is something that we put on the back burner after a grant was rejected to pursue this, because there had been a lot of controversy in the literature that CMV is actually present in the tumors. So we haven't pursued that, but we are now reinvigorated to pursue it because a colleague in Sweden has now developed a technique to pull out the viral genome, and it indicates that there may be a mutant or sort of aberrant viral genome associated with the glioblastomas. Very preliminary, but it would suggest that maybe some of the controversy in the literature is due to the fact that we're not dealing with the standard root viral infection that we know as CMV. Regardless, what our goal is along this path is to understand which viral proteins are present in glioblastomas, and if they're associated with a mutant virus or aberrant strain, try to identify these. And then based on work that was done by some colleagues at Duke, John Sampson, they took our early findings and have developed a dendritic cell vaccine and have published 2 papers with, I would say, pretty promising results. The one they published just last year indicates that when they vaccinate against the viral PP65 protein, which we found in about 2/3 of our patients, they got just in a very small number of patients, so it's probably not a major statistical significance. Nevertheless, I think now at about 6 years, they have 4 of the 11 who are still alive, which is pretty striking. So what our hope is to perform further studies to see if this viral association can promote the glioblastoma tumor stem cells, and then maybe develop vaccines against IE1 and other viral proteins that may not only cause an immune response against the tumor, but also attack the tumor stem cells that seem to be harboring more virus, we believe, than some of the other cells in the tumor as a strategy. So that's kind of the first part of the 3. So I'm going to move now to the totally unrelated projects that are ongoing now, and Derek is sitting here, and he was speaking earlier. He's one of our patients on a high throughput drug screening project that was started by my colleague Greg Foltz, who was here at the Ivy Center and recruited me here several years ago. And this project was supported by Catherine Ivy Foundation, and Parvinder Hothi is the main investigator here in the group that has been doing the basic component of this in the clinical aspect. But you've all heard the concept that there is a concept we were just talking about, that treating tumors with standard treatments may kill some of the proliferating tumor. Maybe this is not a good model, but a group of resistant cells that may have stem cell properties to a certain extent can repopulate the tumor. So the concept was, what if we could use the modern technological advances of high throughput robotics and drug screening to grow up millions of tumor stem cells from a given patient, and then screen them through hundreds or thousands of compounds, and then come up with FDA-approved compounds based on certain criteria that could penetrate the blood-brain barrier and be used as therapies. So basically what we do is when I resect a tumor, we dissociate the tumor, put it in stem cell conditions, either on laminin or in neurosphere conditions. And then once we've had 10 million or so of these cells grow up, we put a certain number of cells of these tumor-like stem cells into 384 well plates, and we run them through a robot that exposes 8 drug concentrations of different drugs to the stem cells. And we've done this many times with normal neuronal stem cells as sort of a toxicity control. And then you let them grow, and then you record the viability of the cells after a few days, and you basically see which drugs are toxic to the stem cells. And the early studies that were done by Greg Foltz and Parvi showed that there was a lot of variability from patient to patient. Here you see 3 patients where these cells did not respond at all, and these 2 patients' tumors responded dramatically at a certain concentration for whatever drug. And one of the drugs that was really kind of interesting that came out of this screen as a potent tumor stem cell inhibitor was Disulfiram, which is a drug used to block, or it's been around forever for treating people who are alcoholic to prevent them from drinking because they get a bad reaction. And it turns out that it's aldehyde dehydrogenase inhibitor, and aldehyde dehydrogenase is a hallmark of, at least in breast cancer stem cells. So these preliminary studies led to a phase 1 clinical trial with the objective of feasibility and safety of giving patients a 3-drug cocktail specific to their own tumor, glioblastoma stem cells toxicity screen. So we started enrollment in December 2015, and to date 25 patients have been enrolled, at least in the collection phase. The drugs are given if they have a recurrent tumor at the time of recurrence, if they qualify in terms of various qualifications, Karnofsky performance score, et cetera. I've essentially told you all this, but it's pretty sophisticated in terms of how we have a oncology pharmaceutical group that goes through all the drug-drug interactions and a data safety monitoring board, and then the neuro-oncologists have to put their okay on it. And once these have been okay, they can prescribe the drugs, as in the case of Derek here, who's on 3 drugs. And the patient's drugs that have been selected have come from all kinds of different categories: anti-neoplastics, anti-infectives, antipsychotics. Zoloft is one, as Derek was saying, that happened to be, for whatever reason, potent against his tumor. And so these are the results of the 4 patients so far, just for one drug. I'm not showing the name of the drug because it doesn't really matter, but are 2 different drugs where the 4 patients, in this case, the blue patient here had a more sensitive response to this drug, and in this case, this patient had a dramatically more sensitive tumor stem cell response to this drug, and that drug was one of the ones picked for their treatment. So we've treated 4 patients now, including number 4, who's in progress right here. And so far, we've had no significant side effects or safety issues. Obviously, it's too preliminary to make any claims. And each of these patients are on different combinations, so you can't say the therapy works because everyone's getting something different. Nevertheless, for instance, in patient number 3, we had, after, I guess, 1 month, a pretty interesting MRI response. This patient had recurrent glioblastoma that was multifocal, and they came back in both hemispheres and in the cerebellum, and all of the lesions became significantly smaller after 1 month of her cocktail therapy that was specific to her tumor stem cells. And this is just an example of one of the lesions after 1 month went from this size of enhancing lesion to that, and there was no other drug like Avastin or anything involved at this point. So we're hopeful that this will lead to some new insights. Obviously, this is very crude. It's a safety study, but the end goal is to expand this trial and then work with people like Nitin Baliga, who was here earlier today at the ISB, to try to reverse engineer why certain patients are responding to certain drugs and maybe see if that has something to do with the genetics or biology of their tumor. And hopefully down the road, this will allow us to figure out certain pathways that could be targeted in these tumors. The last study was also envisioned by Greg Foltz and Parvi, and it's in the preclinical stage. And this is a personalized RNA-based dendritic cell immunotherapy approach to GBM cancer stem cells. So in this case, just like the previous iteration, we take the patient's tumor cells, we grow them in stem cell conditions, but at that stage, we take the tumor cells and extract the RNA with the concept or hope that the RNA expressed by the patient's tumor or glioblastoma stem cells may contain transcripts that would encode for antigens that may be somehow specific for that patient's glioblastoma stem cells, or could make them more vulnerable to immune targeting. So we have a phase 1 trial that we're working on the preclinical component for, and we, for reasons similar to other investigators, we picked dendritic cells because they're most effective at antigen presentation. The goal will be to take the tumor biopsy from the patient and grow the tumor stem cells, and then take the RNA from these, and then simultaneously take the patient's, do a leukapheresis and isolate immature dendritic cells and pulse these dendritic cells with the RNA from the patient's own glioblastoma tumor stem cells. It's somewhat analogous to the DC VAC study, somewhat analogous, because this study, which we were one of the biggest participants in, essentially took a tumor from a patient and took the proteins from that tumor and then exposed their patient's own dendritic cells to the tumor proteins. This is a little more selective just to the tumor stem-like cells, not the whole protein gamish from the tumor. There is some preliminary evidence that this may be a viable way of approaching this tumor. There was a study out of the Oslo University Hospital that was published a few years ago that was a very similar study after, and you can see here, therapeutic vaccination against autologous cancer stem cells. In this case, essentially the same algorithm was utilized. Patients gave tumor biopsy. The RNA was then amplified and then pulsed into their dendritic cells and given back, and then given as vaccine at various time points, as you can see here, throughout the course of their between radiation temozolomide and between temozolomide treatments. There was pretty good preliminary evidence, even though it was a small study of 11 patients, that there was some improved efficacy for progression-free survival from a control group from about 300 days to a median of about 600 days. I'm not sure or aware of what the follow-up has been with this group. Our preclinical research project was already underway when this was published, so it does kind of look like we're doing a Me Too, but we've made ours into a 2-arm clinical trial because in the intervening time, checkpoint inhibitors have come out, and we believe that if we did this 1 arm with just the dendritic cell vaccine with the RNA from the tumor stem cells, and then another arm with in the presence of a checkpoint inhibitor, that we might be able to enhance the immune response to these patients. So this is a work in progress. We're just at the preclinical stage getting approvals from the FDA, and hopefully we'll be able to move this into patients in the next year or so. So as shown here, that'll involve surgery, leukapheresis, and then radiation and Timidar, just like the study from Norway, and then 2 arms with or without checkpoint inhibitor. And this basically just talks about the same thing, that we isolate the dendritic cells from the patient, how we isolate the RNA from the tumor, and then use an electroporator to pulse that RNA into the dendritic cells. The regulatory components have been achieved for the most part. We are working with a clean room over at the University of Washington that we're able to rent, really nice facility there. Following these approvals, we will hopefully be enrolling patients soon. So I have a lot of people to thank, and I don't have their pictures or their names here. I've been a beneficiary of coming to an institution that had a lot of ongoing projects. Another of our investigators, Ralph Pachowski, is here, and he's doing some wonderful work as well. And our whole group is fortunate that we've had the funding from the Ivy group. Hopefully some of these clinical trials will lead to more insights as to how we can attack these tumor stem cells. And in the meantime, Peter's probably going to figure out much more elegant approaches with rational drug design. But I appreciate you all coming, and I think that it was a really wonderful event, and bringing together some of the top minds in the country for the cutting edge research that needs to be pursued for glioblastoma and brain tumor treatment. Thank you all very much again.